Hello, welcome to today's lesson. We are going to dive into PICO framework for research. We are going to look on how you are supposed to formulate a research question, and focusing on the PICO framework, we are going to discuss what it is, and we are going also to look at the examples that I'll be showing you, and then therefore you are good to go and form your own research question based on your field of interest. Welcome. So to begin, what is PICO in full? So PICO is an abbreviation that stands for P for population, I for intervention, C for comparison, and O for outcome. So usually when we are doing research, or when you want to do a particular research in a particular field, the first thing you need to know is your population. What questions do you have about a particular population that you want to study? Again, secondly, what do you want to do as part of your research? So that becomes your intervention. And because already we have existing systems, so whatever you're bringing in as new, maybe in quotes, you will be comparing it with the existing or maybe others. And then we have the outcome, that is now the result, what you think will be the outcome of your intervention. We have different types of population, and for example, those that are delving in medical research, biomedical research, most of the time you'll find that our population is a particular group of patients, maybe with a particular disease. For example, you can have adults who are infected with HIV as your study population. We can have children, maybe between this particular age to this particular age, who are living with maybe sickle cell anemia or any other condition. So population is basically a group of people with a particular characteristic that you're going to study. But usually you're not, because of resources, you may not be able to study the entire population, and that's when you'll just do a sample of your entire population. So before you come up with a question, you must be having a population in mind that you want to study. For example, we are using this case, adults who are having HIV. And then we said we have I that stands for intervention. Now, if for example you were studying people, adults in this case, who have been infected with HIV, now what's your intervention, or what do you want to bring on board? I know we are going to talk about the entire formulation of that particular question, but I'm bringing up I'm trying to define the PICO framework first, and then we will delve into better examples. So the intervention is the exact thing that you're bringing in on board. For example, it could be a new treatment. We already have some existing treatments, and now you're bringing in something new, or you're trying to test something that hasn't been tested yet. So that will be the intervention, where you come in, the exact action you're going to take during your study. And then we have I said PICO, P for population, I for intervention, and then we have the comparison, that is C. So for example, I could be studying patients who are adult patients who are HIV positive, and they are being treated using a standard drug. And then my intervention is to test a new drug and find out if there is something different. So I will be at this point comparing two particular drugs, the one that I'm bringing in as my intervention and the other one that is existing. So we call this a standard drug. in this example. And then we have the outcome. They are always the results you anticipate. If you're bringing something new. For example, you will expect to see a viral suppression, maybe compared, you are trying to test drug A, if it brings better, if there is an improvement when it comes to viral suppression, sometimes reduced mortality. So outcomes are generally the results or the outcomes from that particular test that you will be doing. So this is an example of a full PICO question. In adults with HIV, does DTG, that is an antivirus, compared to efavirenz, that is another antiviral, lead to better viral suppression? So that is an example of a research question that has been formulated based on the PICO framework. So in this case, you are able to see that we are talking about adults who are infected with HIV. So that is the particular population I'm targeting. And then I'm looking at this particular drug here, the DTG antiviral. That is the intervention. That is what I'm trying to test or bring on board. And then I'm comparing this DTG, that is my intervention, with the standard treatment. For example, in this case, it's efavirenz. And then I'm checking whether the use of DTG can result to better viral suppression. So that will be the outcome. So when you're formulating a research question based on PICO, you will be addressing, number one, your research question must have a clearly defined population, clearly stated your intervention, and then your comparator, and then the outcome. So let's look at more examples. We have an example here of a research question that I want us to look at it and then dissect it and find where is the P, where is the I, where is the C and O. So this is a question: in patients suspected of bloodstream infection, does metagenomic next generation sequencing compared to conventional blood cultures lead to faster pathogen detection and improved treatment accuracy? So number one, we say the population is that particular group with a specified characteristic. So in this case, the patients who are suspected to have bloodstream infection. So this is the population that you are going to study. And then, now that is the P, we are looking for the I. Our intervention here is the metagenomic next generation sequencing. That is something new we are trying to bring on board. And then we are comparing it with the standard method, which is the conventional blood culture that is in use. And then our outcome, we are trying to see whether it will be able to bring a faster pathogen detection, that is first outcome. And in this case, we have two outcomes. So we are looking at the rate of detection. Is it faster? And then also, is improvement treatment. We're also looking at the improved treatment accuracy. So we are looking at accuracy. Is it better than the conventional blood cultures? That is, if we are going to use metagenomic sequencing. So this is an example of a good, a well-formulated research question based on PICO framework. There we go. So this is it. The patients suspected of bloodstream infection are our population. Secondly, our intervention is the metagenomic next generation sequencing, which is a type of a diagnosis. And we are comparing it with the other type of diagnosis, that is the conventional blood culture method. And the outcome is we're checking whether there is faster pathogen identification and improved treatment accuracy. Let's look at another good example of a well-formulated research question. In hospitalized patients with multi-drug resistant infections, does whole genome sequencing guided therapy compared to empirical antibiotic therapy reduce treatment failure? So the kind of population that we are targeting here, we are looking for, number one, patients who are hospitalized, not just patients who are maybe they're sick and at home, or maybe they're outpatient. We're talking of hospitalized patients. That is the first characteristic of this particular group. We say the population is well characterized. So here in this case, the first characteristic of our population is that the patients are hospitalized. And the second thing about this particular group, we are targeting on those that are having multi-drug resistant infections. So looking at this carefully, we will, number one, if we are to do an inclusion or exclusion criteria, the first thing we will exclude patients who are on outpatient treatment, that is, or patients who are outside the hospital. We will exclude them from the study. And number two, while we are the hospital, we will still exclude those who are hospitalized, yes, but then they're not having multi-drug resistant infections. So we are very specific on the kind of population that we want to study. Those that are hospitalized and infected with multi-drug resistant pathogens. And then what is our intervention? It is still on whole genome sequencing guided therapy. And then the comparator is the empirical antibiotic therapy. And then our outcome, our result is we are trying to see whether we will find a reduced treatment failure. So that it is hospitalized patients with multi-drug resistant bacterial pathogens are our population. And as we said, the intervention is what we are doing, what we are trying to bring upon the existing. And then the comparator is whatever is already there, or sometimes a control. Maybe we can go with a completely no therapy, and then or reduced mortality or treatment failure as our outcome. Thank you so much. Let's meet on our next video as we dig deeper into the field of research. Thank you.